DNA in school. However, we were not satisfied with only knowing about the basics, knowledge of DNA. So we took matters into our own hands and did some research. We knew that DNA is very important and figured that if we were able to figure out which section of the DNA sequences are in charge of what, for example, cancer, it could greatly benefit our society, the medical fields, and the agriculture of our country. But then a question struck us. If DNA sequencing is so important and can help our community, why is no one doing it on a large scale? Well, although it is very beneficial, it is tough. DNA sequences are very long and complicated, so it would require experts to do the job. Because it is so long and complicated, it is time consuming. And with humans being humans, we make lots of mistakes. And one mistake when sequencing DNA could turn the outcome into a whole different thing. Then we thought, if we can't do it, why not let someone else do it? Someone who can do it fast and accurate. We came up with the solution of AI. This is our mascot. Yeah. As we learned about AI, we came to know that there was a field in AI called natural language processing, which specializes in giving the computer the ability to understand human text and language, which can then be used to classify text and carry out multiple functions. We looked at the DNA sequences and realized that the sequences look like human languages. So we decided to treat DNA sequences like human language and use the NLP to classify it into gene families. During the research, a couple of analogies were introduced to explain AI information and concepts understandably. They were displayed using interesting images to give us a better understanding, and we have successfully achieved our goal of auto-recognition of DNA sequences. This is our project plan. So first we gathered the DNA sequences, transformed them into a human language, and we used the concept of vectorization to turn it into a language that the uh, computer can understand. Then we ran it through the AI models and classified it as ion channels. We began by first collecting the data and transforming the language. The figure shows the transformation of DNA sequences into a text we can understand. They are composed of the letters A, C, G, and T in particular orders. Yeah. To have the computer understand and classify these sequences, we needed to split the entire DNA sequence into words and phrases. We keep each word to 4 characters and have them overlap like how the diagram shows. A couple of certain amount of words for a phrase. This is the math representation of the process and transformation. And now we have transformed the DNA sequences into something similar to human language. The next step in our classifying process is to transform the words into languages that the computer can understand. In other words, vectorization. As the diagram shows, the computer can't understand human language. So we use tools to transform it into numerical language for the computer to understand. There are 2 methods we chose to experiment with, which is the count vectorizer and TF-IDF, because they have the highest performing abilities. But before we dive into the 2 methods, we had some questions. For example, why vectors are included in this project? Well, we knew that vectors are an attribute in math, has magnitude and direction, and reflects the last slide and how it could be used for our project. For example, how many times does this word appear in this text and such? It can also be understood as the math graphs we learned in school, which had a x-axis and a y-axis. The magnitude of a variable is its direction, whether it's increasing or decreasing. This is important so that we can let the computer classify the DNA sequences by asking how many times a sequence appears and what this means. First comes the count vectorizer. It not only transforms the words into numerical language, it also counts up the number of times this word appears and lists it. This is represented in mathematical equations over there. Yeah. Our second vectorization method is the TF-IDF. It is a slightly more complicated and thus theoretically better. It not only transforms the words and counts their frequency, but also compares it to how many times they appeared in other texts it has in its independent dictionary. So unlike the count vectorizer, the numbers appear as decimals. This is the formula we represented in, like the math one. Yeah. And supervised learning is also something we included in our project, and it can be seen like in the diagram. Like we couldn't find a way that was understandable for us to learn this. So at the start, so we used analogies to help. We understood supervised learning, seeing it as an analogy of a baby learning with his parents supervising them. The trained model is like the adult who is able to make their own decision based on the patterns it learned as a child. The diagram on my right indicates the biological nerves in a human body and brain. Oh, well, on my left, yeah. On my right is the diagram of nerves used in AI. The red line and the green lines are like how the synapse determines the weights, and this can be taken in as the input of a flick and a punch. The output is like how your brain reacts to both. Your brain will know that the flick was light and the punch was hard. Once the weight enters the blue blob, the bias will keep it balanced, and the output for every node can be described with the formula as follows. In this formula, bias is the constant value, with f being the activation function. But what is an activation function? Not all neural nodes of the model are used. Some remain unused. So how do we decide which nodes to activate to maximize the accuracy of the output? And what if the numbers are too great? Well, activation functions create a range. Everything below this range is small. All above is big. What's done in between is not so important. Yeah. And we chose these three. The training model will answer the question, which is the prediction. Then it will be given a correct answer, the true answer. The loss function is the chasm between the true answer and the prediction. The optimizer would then make the output go back into the hidden layer and try again. The training model will try again and again until the answer is correct. And yeah, as we had mentioned, the loss function is the difference between the prediction and the true answer, and the optimizer minimizes the loss function. There are lots of loss functions and optimizers, but in this project, we use the categorical cross-entropy loss function. And for the optimizer, we used Atom because it was like considered as one of the best ones. And we may explore the others in future projects. These are some of our data sets. We included the concept of generalization in our project, meaning that this project can be used for multiple species, like as seen as like the human, dog, and chimpanzee. We experimented and found that putting 4 characters in a DNA sequence into each word would result in the highest accuracies. An interesting finding is that the accuracies were slightly lower with TF-IDF, though a better performance was expected from theory analysis and experiments and some NLP references. A possible reason is that the words in our projects are not actually words, so it would not be very helpful to introduce IDF. Moving on, the tables show the results of using different activation functions. As you can see, ReLU had the highest accuracy, and so we used it for our models. This is our results in matrix form. There is a high accuracy for classification of label 6 and low accuracy for class label 5. This is probably because of the difference in amount of datasets of each class as shown on slide 17. As you can see, this is our history for APOCs and accuracies. Humans had the highest, which wasn't that fascinating. And what was good was that in the end, well, they all reached quite a high accuracy and pretty stable before the APOCs reached 100, which is pretty good. And as mentioned earlier, we included the concept of generalization from transform learning. Generalization is very important in recent days. We need to save resources, so we need to make a model that can carry out many things. And yeah, this is our summary. We learned to balance in real-world applications. When we decided on experiments and network settings, we took into account its performance and accuracy against the cost of time and computer resources to find something that is accurate and efficient for experiments and networks. And secondly, AI is a data-driven tech. And as it was seen in previous experiments, the classes with higher amounts of data achieved higher accuracy, and the classes with less data achieved less accuracy. And the third is the method used depends on the project. This can be seen from our choice of vectorization, which TF-IDF did not turn out as the better result for our project, which was theoretically supposed to be better. And fourthly, the biology connections can be reflected from the patterns. Yeah, through the generalization projects experiments, we can see that two species had more similarities in terms of their DNA. Yeah, but all in all, we completed this project and made a demo, papers, and presentation. We added creativity into our ideas and complete the recognition of DNA sequences by transferring them into human-like language using NLP. Our project was capable of being used on 3 species, and this project could benefit the medical fields, scientific fields, and many more. In the future, we will be working on making this project capable of classifying DNA during mutations. Yeah, thank you for your time. Any questions?